So what is CRISPR? It's a tool that scientists can use to edit genes. Not those genes, these genes. The acronym stands for Clustered Regularly Interspaced Short Palindromic Repeats. So how exactly does CRISPR edit genes? One analogy I like to use is that of a Swiss Army knife.
CRISPR can be used to cut, alter, rearrange, or add in new DNA.
Though CRISPR may seem to be a new technology, it's actually not. It's a natural mechanism that bacteria use to protect themselves against viruses. It works by making a molecular machine that recognizes viruses and cuts their DNA in specific spots that hopefully kills or at least disables a virus. Scientists have learned how to harness this and use it precisely. What do you think the most deadly animal in the world is? Would it be the great white shark? The box jellyfish? Actually, it's the mosquito. The reason mosquitoes are
This deadly because they carry malaria and many other diseases. CRISPR can be used to fix that.
Another issue is the issue of organ donation. Organ donation itself is a wonderful thing, but every year people die on the transplant waiting list. There just simply aren't enough organs to go around. This could be remedied easily by using one of our favorite porky friends. Human organs are almost exactly the same as pig organs, believe it or not. The reason we haven't used them before, though, is they're actually riddled with viruses, known as porcine endogenous retroviruses, which are similar to the AIDS virus. For obvious reasons, no transplant applicant would want an infected organ, and no respectable donor organization would conduct transplants with diseased organs. However, CRISPR technology could be the key to ridding the pigs of the viruses. With these systems, researchers can permanently modify genes in living cells and organisms, and may eventually make it possible to correct mutations at precise locations in the human genome in order to treat genetic causes of disease. So CRISPR can be used to manage or hopefully even get rid of genetic diseases. This is all theoretical right now, as scientists haven't done much with either stem cells or human embryos. But let's say that science does get to that point. Stacy here has sickle cell anemia. If scientists manage to cure some or all of her symptoms, or maybe even the disease itself, that would be a success. But there is another consideration. To accomplish this, scientists would have to edit her genome to get rid of the DNA that causes the disease. But your genome is almost a living part of you, and you pass it down to your children if you have any. So let's imagine that she does have children, and then her children have children, and this goes on for generations.
No matter how far down the line you go, there will always be a possibility of the altered gene resurfacing.
And with that
possibility
comes, well, we don't even know. This is all uncharted territory, and it's unlikely that we'll be unpacking it anytime soon, probably not even in our lifetimes. With all of these ideas, research, and experiments, each scientist must restrain themselves to one thing: ethics. Ethical complications are never more numerous than when you're talking about editing human DNA. With all of these considerations, why is CRISPR worth it? Why is this so widely utilized? Well, it's the most common method, and it's most reasonably priced as well. There are the infinite possibilities that you can use CRISPR for, and it's also the speediest and most precise at recombining DNA. All in all, CRISPR technology and the entire science of recombining our genetics may be the most power humans have ever had over nature, and it's imperative that we wield it responsibly.